Hello, welcome to my channel Deep Learning Biology. My name is Dr. Vikas Bhardwaj, and here today I am going to teach you about enzymes. So here within this class we are going to have a discussion related to enzymes. Now here I just want to tell you, if you look into the history, earlier the enzymes, they were being isolated from yeast. Hopefully you must be knowing, that yeast, it is widely used for the production of ethanol. Now earlier scientists, they used to believe that by using yeast, the complete yeast, only you can produce ethanol. But here I just want to tell you, later scientists, they isolated from the yeast proteins, which can catalyze the production of ethanol. So here, hopefully you have learned in your classes the general term, this enzyme means from the yeast. Now keep in mind, enzymes, these are actually the proteins. Enzymes mostly are proteins which can carry out biological work within the cell. Hopefully you have attended my previous lectures. In all these lectures I have told you, within the cell, you are going to have 5 major type of biochemical reactions, and hopefully you know that these biochemical reactions, they can be categorized in 2 categories. One is catabolic reactions, another is anabolic reactions. Now all these reactions which are occurring within the cell, which are occurring within the living cell, they are occurring due to the presence of enzymes. Now here I have told you that most of the enzymes, they are proteins, but I also want to tell you some RNA, it can also act like enzymes. And these catalytic RNA means the RNA which is going to have the property of enzymes, they are known as ribozymes. So this is the introduction about the enzymes. Now here I just want to tell you that if you see any bacteria, if you see any bacteria, within the bacteria you are going to have enzymes, and all these enzymes which are present within the bacteria, they are carrying out diverse activities. They are carrying out biological work within the bacteria. Now here I just want to tell you, I am going to take one example. Hopefully you know, generally the bacteria E. coli, it is going to have a life of 20 minutes. Just 20 minutes life. Now in this interval, it has to carry out all the things. Here I just want to give you one example. Within the bacteria you are going to have one enzyme, OMP decarboxylase. This enzyme is responsible for synthesis of pyrimidines. Hopefully you remember purines and pyrimidines. This enzyme, OMP decarboxylase, it is responsible for synthesis of pyrimidine, thymine, cytosine, uracil, and later these will be incorporated within RNA, or you can say DNA. Now here I just want to tell you, OMP decarboxylase, it is going to carry out the synthesis of pyrimidine, the required amount of pyrimidine which bacteria require in approximately 20 milliseconds. Now, the same reaction, if you are going to carry out, okay, it will take 78 millions years. Means, try to see if the reaction which OMP decarboxylase enzyme is doing within the bacteria, okay, it can carry out within 20 milliseconds. If the same reaction you are carrying out outside of the bacteria in vitro, it will take 78 millions years. Now, hopefully, you are going to see that the enzymes, okay, what they do, they accelerate, they increase the rate of reactions. This is the most important. Keep in mind, enzymes, they are biocatalyst. Hopefully you know what is a catalyst. A catalyst, what it does, it speed up reactions. Hopefully you must be knowing in car, okay, what we are going to see, the fuel, it is being burned, okay, then you see the pollutant, they are being released, okay. Now within the car you are going to have catalytic converter, okay, there is a catalytic converter, okay, and that catalytic converter will convert the pollutant into less toxic substance. Now here you must be knowing what are catalysts. These catalysts, they are going to take part in the reaction, but they are not being consumed. Generally the catalysts, they speed up chemical reaction. But here you see the enzymes, they are biocatalyst, okay, they also increase the rate of reaction. Now here I just want to tell you that let us suppose you are going to have enzyme, okay, you know very well it will combine with the substrate. What we are going to see, you are going to see formation of enzyme substrate complex, then you are going to see the formation of enzyme product complex, and later what we are going to see, enzyme will be released and the product will be released. Now here if you see this reaction, okay, this reaction is occurring both in forward direction as well as in reverse direction, okay. So you are going to see that enzyme, okay, the enzymes, okay, what they are doing, they are converting substrate into product and product into substrate. So keep in mind, if you are going to have any enzyme, what it can do, it can convert substrate into product and product into substrate. So here you can remember that enzymes, what they do, they carry out interconversion of substrate and product. Now hopefully you remember what is chemical equilibrium, okay, chemical equilibrium. Now here if you are going to have a reaction, okay, let's suppose substrate, it is being converted into product, product, it is being converted into substrate. Let us suppose you are going to have initially you are going to have 100% substrate, 100% product, okay. Let's suppose you are going to have 1 molar concentration of substrate, 1 molar concentration of product, okay. Now let's suppose you are carrying out this reaction outside, okay, and what is going to happen, let's suppose this reaction in 1 hour it is going to reach the stage of equilibrium. At equilibrium, what you are going to have, 70% as and 30% product. It may be anything. Okay, so for different reaction you are going to have different, and here you can also calculate the equilibrium constant. Now here, in presence of enzyme, this reaction, okay, this reaction, it is going to occur very fast. It is going to occur in millisecond. Okay, so here keep in mind the enzymes. Okay, what they do, they enhance the rate of reaction. Okay, yes, the enzymes, if they are present within the system, okay, the equilibrium point, okay, will be reached faster. Okay, but keep in mind enzymes, they do not change the equilibrium point. Try to see here, in this reaction at equilibrium, what you are going to have, 70% S, 30% P. Okay, now here in presence of enzyme, this reaction will occur at a fast rate. In absence of enzyme, it will occur very slowly. It may be 1 hour, 2 days, whatever it is. Okay, so hopefully you are going to remember enzymes, they are going to enhance the rate of reaction. Yes, in presence of enzymes, equilibrium point will be reached at a faster rate. Yes, keep in mind the enzymes, they do not change the equilibrium point. So hopefully now you are going to remember that enzymes, okay, they enhance the rate of reaction, okay, and they are going to enhance both forward reaction as well as they are going to enhance the backward reaction. Yes, I have told you that enzymes, they are, one minute please, proteins. Okay, and now every protein is going to have a life. Okay, it is also going to have a life, that it will be synthesized, later it will be degraded. Okay, yes, keep in mind that enzymes, they are going to enhance the rate of reaction by 10 to the power 6 to 10 to the power 17 times. Another important point, keep in mind the enzymes, they are going to work most of the times in mild condition. Okay, yes, most of your enzymes within the cell, they are going to work at pH 7 and they are going to work at the temperature that is 37 degrees Celsius. Okay, yes, here I also want to tell you that enzymes, generally they are required at less concentration. Okay, it's a small concentration of enzyme, it can carry out the reaction. Okay, and finally the most important thing, the enzymes, they work with specificity. Okay, they are going to work specifically. Okay, so these are some of the important points. Now here I just want to tell you, if you see the protein which are going to work like an enzyme, so this is the protein. Okay, so here this is the protein which is going to act like enzyme. So here you are going to have a pocket, okay, within the enzyme. Okay, this is known as active site. Okay, at the active site, what is going to happen, substrate will come and bind and later it will be converted into product. Now in the next class I am going to teach you how enzymes, they actually work. Okay, so this point we are going to discuss in the next class. Thank you for watching this. Please do like, share the video and subscribe to my channel Deep Learning Biology. Hopefully you are enjoying it. Thank you.